Okay. Microbiology can be divided in different fields. So if we are talking about different types of microorganisms, we can divide microbiology in mainly in three main groups: bacteriology, virology, and mycology. How do you think which microorganisms or which pathogens are the mostly predominant in this world? Bacteria, viruses, or fungi? Your opinion. Who is for bacteria? Who is for viruses? Who is for fungi? Who is lazy and doesn't want to participate? And who is not listening? Okay. So, how do you think? Which type of microorganisms are the mostly predominant in this world? Huh? I need to see the opinion of majority. Till now, just minority participated. You think so just because today we are talking about fungi? Yes? No? So how do you think? I need a loud... did you Google? Well, first of all, I guess the mostly correct answer that science is not aware yet which one is the mostly common, for a few reasons. We didn't discover more than we did. So maybe now we have opinion that some of them are more common in the nature. After them we will discover more. Today we are talking that viruses are mostly common. Tomorrow we will discover that bacteria are we have more bacteria in amount, and after that we will discover that fungi. But basically, according to the data we have right now, anyways, viruses, we have more viruses, as well as we know, as well as we could appreciate right now. And it makes more sense, at least because of the fact viruses can infect any type of cells. There are some viruses can affect plants, there are some viruses can affect fungi, there are some viruses can affect animals and humans. So basically for each cell, for each living organism, tissue, microorganism, cell, there is a virus. There is appropriate virus that could infect this microorganism, this microorganism. This is why probably we have more viruses in nature. On the second place, bacteria, as well as we know. And on the last place, we have fungi. Meanwhile, we have very large variety of fungi. And by talking about large variety of fungi, we can make difference, for example, between macrofungi and microfungi. Yes? Do you eat mushrooms? Yes. Do you like mushrooms? Yes. Of course. But hopefully we are not eating candidas or something like that. So, fungi are extremely interesting from different points of view. They're uptaking very large space. They are making their own kingdom. Think about it. They have their own kingdom. Even humans are not a separate kingdom. Humans are making part of kingdom of animals. So we are animals with wisdom, some of us. Yes? And with judge, some of us. Maybe, hopefully. But even humans are making part of kingdom of animals, while fungi are a separate kingdom, among which we have both microorganisms and macroorganisms. If we are talking about mushrooms, mushrooms are macroorganisms. We can see them, we can touch them, we can consume some of them, or we can just simply admire some of them, but you better don't touch them. There are some fungi that are multicellular. There are some fungi that are unicellular. If we're talking about bacteria, they are just unicellular. We don't have any multicellular bacteria, right? Meanwhile, bacteria are making part of domain of prokaryotes, and fungi are making part of domain of eukaryotes. Do you remember the difference between the cellular structure of both of them? We'll discuss it a little bit later. But it is very interesting type of organisms. What is interesting about them except their variety? Here you can see basically how can they look, but actually you know how they look. So also they play different roles. Some of them are playing roles in food industry, some of them are playing role in pharmaceutical industry, right? We have some fungi that are participating in antibiotic production. The first one, the mostly known one, is Penicillium. Penicillium is the name of fungus, and the name of antibiotic is penicillin, that comes from penicillium. Also, they're making part of, they are useful in biotechnology by producing different enzymes. And by the way, they're making part of normal flora of some animals, because they produce some enzymes that are participating in the digestion of some animals. Just as we have as normal flora bacteria in the intestinal tract. So bacteria also helps us to digest some elements. And of course, one of the mostly important roles of fungi is circulation of micro and macro elements in nature. That is known as saprophytic function. Whenever we are talking about saprophytes, it is not just about bacteria. It is also about viruses. So whenever you are thinking about decaying materials in nature, about circulation of elements in nature, the mostly useful, the mostly important participants of these processes are saprophytic bacteria and fungi. And here we are talking not just about microorganisms, but also about microorganisms. Did you ever go to a forest to take some mushrooms, to harvest some mushrooms? Did you ever? At least I'm sure you saw how they're growing, right? And some of them are growing, the mushrooms we can consume are growing specifically nearby specific trees, nearby specific bushes, nearby specific in the specific area. So what does it mean? They're consuming specific micro and macro elements. They are also making ensuring this process of circulation of elements. But some of them are consuming just good materials, just useful materials we can consume. And some of them are consuming basically the toxic matter, or produce toxic matter while their metabolic process. In the kingdom of fungi, we have different phyla. And these phyla also are in a big variety. So whenever we are talking about taxonomy of fungi, whenever we are talking about their naming, their nomenclature, it is also important to differentiate them and to understand from which part, from which group, from which phylum they are making part, because since they are different in shape, since they are different in structure, in metabolism, they will have also different way of multiplication. They will have also different different places they will affect. Of course, the mostly important fungi for us from medical point of view, on the first place, are pathogenic ones. We're not talking about fungi that are used in the food production or in pharmaceutical production or biotechnologies, because this is the field. It is not field of doctors. Doctors should be mostly interested in pathogens. Pharmaceutical companies will produce drugs, but we are interested about pathogens. So whenever we are talking about investigation of infections caused by fungi, what should we take into consideration? In the fungal diagnosis, in the fungal lab investigation, you will observe a lot of things that are in common with bacteriology. Very often same tools are applied in bacteriology will be applied in mycology. Same methods of microscopy, same methods of serological investigation, same methods of cultivation can be applied in a lot of cases in mycology. So even sometimes it is very hard to differentiate on the culture media, is it bacteria or it is fungus. How we make difference? Because if you remember, there are some bacteria that are looking similar under the microscope, very similar to fungi. For example, actinomycetes. Actinomycetes are bacteria, but they are looking similar just as some fungi. On the other side, some fungi are looking similar with bacteria because they are unicellular without any filaments. So, how we make difference? First of all, we keep in mind that they are making part of different domains. So, a lot of bacteria are looking like fungi because they're having some filaments, and a lot of fungi are looking like bacteria because they are unicellular. So how we differentiate them? Especially if we are using such tools like electron microscope, that gives us a bigger possibility for detailed diagnosis, for detailed studying. You will observe that some of them are prokaryotic cells and some of them are eukaryotic cells. This is the main thing that makes them to be different. pro and eukaryotic cells. That means when we are talking about bacterial cell, you can see cell wall, you can see nucleoid, that is main part of genetical material. We can see plasmid, a circular DNA that is extra genetical material, some ribosomes, cytoplasm, cell wall. It is about prokaryotic cell. When we are talking about fungal cell, that is eukaryotic cell, here we have nucleus, here we have Golgi apparatus, here we have mitochondria, vacuoles, and so on. So we have eukaryotic cell that has organella that is characteristic for all eukaryotic cells, including plants and animals. So otherwise, if they wouldn't make part of different domains, we wouldn't be able to make difference between them. Now, if we are talking about way of multiplication, which is the way bacteria multiplies? Binary division. Which is the way fungi are multiplying? There are a few ways, but the main one is spore forming. Also, we have some bacteria that are spore forming, right? Like bacilli, like Clostridia. But do bacteria multiply by using spores? No. If we are talking about spore forming, spore production by bacteria, it is not a way for multiplication. It is the way for survival of species in nature. So if bacteria will die, the spore will persist in the environment until it will get in comfortable conditions. When it will get in comfortable conditions, it will transform, it will germinate from spore into vegetative form of bacteria and will continue its life. So in this way, genetical material will be preserved until we have comfortable conditions. It is not a way for multiplication, it is a way for survival of species in nature. When we are talking about fungi, that includes micro and macroorganisms, most of them are multiplying by using spores. So the fungus is producing a bud with a spore inside. It falls down or it falls around the fungus, and in comfortable conditions it germinates, it transforms into vegetative form of a fungus and is continuing its life cycle and is reproducing. So in one case, please don't misuse, in one case spore formation is the way of survival, in another case it is the way for multiplication. So we have similar way of investigation, similar but not absolutely same. Because if we are talking about different types of microscopy, yes, we apply light microscopy for both bacteria and fungi. We apply phase contrast microscope for both. We apply fluorescence microscope, confocal laser microscope. We apply electron microscope, and so on. We can apply both of them. To both of them, we can apply all of these types of microscopy. But when we are making the smear, there is a difference between how we make the smear for bacteriological investigation and how we make the smear for mycological investigation. If you remember, while we are making the smear for bacteriological investigation, one of the mostly important parts of investigation, of smear preparation, we prepare the slide, we put there some saline solution, we put there some samples of bacteria, we start spreading it on the slide, and after that what we are doing? Air drying and fixation. Do you remember? We make fixation by using heating. We can also apply chemical fixation, but the mostly common one is physical, with using of flame, with using of heating. So we heat the slide, and bacteria will melt and will stick to the slide. This is how we get the smear of bacteria. When we are talking about fungi, there is a little bit, there is a small difference in smear preparation, and especially in the moment of drying and fixation. We don't apply any fixation. Why? Because the structure of fungi is that fragile, you can damage it by heating, by if we take bacteria, we extend it on the large surface of the slide. If we are taking fungi, what we are doing? We just put a little bit of sample in the middle of the slide. We are waiting for drying, and we cover it with a cover glass. Why it is that important? Because if we will start mixing it like we can do with bacteria, we can destroy these fragile filaments that are produced by most of fungi. As a result, you will see mess. You will see anything but not what you are expecting. So we wouldn't see such beautiful pictures of fungi if we would mess them like bacteria. So we don't apply any fixation, and we apply usually a little bit different methods of staining. Because also they're fragile. That means we will not wash it. We will not. Why we cannot wash it after staining? We just apply a stain. We apply the dye. It is absorbed in the fungal cell, and that's it. We are waiting for drying. Why we are not washing, decolorizing? Because first of all, we didn't apply fixation. If we didn't apply any fixation, what happens? You wash the slide and you wash out the sample with the dye, so you cannot wash it out. This is why a little bit different dyes and techniques will be applied for staining of fungi. So it will depend on samples we are taking, because what is the mostly affected area, which is the mostly affected area if we are talking about fungal infections? It is skin or mucous membranes. Sometimes it is respiratory tract. Less common is bloodstream, but also could happen. But the mostly affected, we have skin and mucous membranes. So here there are two ways. Basically it depends which kind of investigation we want to apply. If we want to apply serological investigation, there is no problem. You can bother the sample just as you wish. Antigens will be captured by antibodies, and you made this serological investigation. But when we are talking about microscopical investigation and cultivation, it is very important to apply very gentle movements in order to see the entire picture, in order to see all the filaments and all the structures. That will confirm a little bit more precise the diagnosis we can apply. Unfortunately, this mycological field in our country is not that common, is not that frequent and developed. Maybe even fortunately, not unfortunately, because usually we are developing parts we need, right? It is like if your car is the common one, it will be easier to find parts for this car. If the infection is common, it will be easier to find the lab that is working on this infection. So on this, from this point of view, that's good. We are not too much developed in mycology, but basically we have a little bit to work in this field in order to provide population with more specific and more proper investigations and more proper treatment. This is all for introduction in mycology. And if you have any questions I could answer, you can ask. Thank you.